Namaste students. I am JBN Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic properties of antigen and properties of antigen antibody reactions. Various kinds of antibody reactions which are part of the properties of antigen antibody reaction. This topic belongs to immunology, immunotechnology, principles of immunology, and basic enzymology. So, antibody properties of an antigen antibody can be explained with understanding the antigen affinity, antigen avidity, and cross reactivity. So these three reactions are there. First, we discuss about the affinity. What is affinity? When an antigen and antibody bind to each other and the strength, the strength will decide what is the affinity of a antibody to a particular kind of antigen. We know that each antibody is specific to a particular kind of antigen. It will not bind to any other antigen, and it will bind to a single type. It will bind to a single type of antigen molecule. So each antigen and antibody reaction is very specific. They are specific in nature. When interaction between antigen and antibody involve some non-covalent binding of antigenic determinants, which are known as the epitope. So each epitope is bound to a particular region complementary determining CDR region of both heavy and light chain. Okay. And these interactions are analogous to those observed in the enzyme substrate reactions, and they can be defined similarly. To describe the strength of the antigen antibody, we can define the affinity. Okay, we can find a term which is known as the term. So suppose that they are loosely bound and they have a weak affinity. Suppose they are bound to each other with high affinity, then suppose this is the antigen and it is an antibody. Suppose they are bound in weakly, then this it may be like this. Suppose they are with high, if they are bound very strongly, then it can say that the affinity is very high. So affinity is the direct term which indicate that the antigen and antibody are bound to each other with high affinity, high strength, and they can be said. It can be said that affinity constant is high. Affinity. What is the formula of the affinity constant? Affinity constant K is equal to antigen antibody divided by antigen multiplied by antigen is equal to 10 to the power 4 10 to the power 7 per mole. So it depends on the antigen and antibody reactions. Now the second term, which is avidity. Avidity is also a term which is related to the strongness of the antigen and antibody complex. So avidity of the antibody, it is the strength of the bond, overall strength. It is the overall strength of an antigen and antibody. Suppose in the affinity only one single antigen was there and one single antibody was there. Okay, but in the avidity, suppose there are 4 to 5 antigens are there, and to those 4 to 5 antigens, 4 to 5 antibody will bind, and their overall strength is calculated. If their overall strength is higher, then it is known as the avidity. The affinity is for the single antigen and single antibody. But avidity is the term which is used for the all of the uh, antigens and all of the antibodies. So if 4 antigens are bound to 4 antibodies, then their overall strength is known as avidity. We can also understand this by the examples of the IgM. IgM is the antibody which has pentameric structure, and it can bind to the 5 to 10 antigens molecule. So if we talk about the avidity, we can IgM is a very good example. If the binding of the IgM to 5 to 10 antigens is very strong, they will bind all the antigens which are present in a single pentameric structure, then this strong binding will be known as the avidity. And only IgG will bind to a single antigen, then it will be known as the affinity. So the avidity is the strength of the bond after formation of an antigen antibody complex. It is used to denote the overall capacity of antibody to combine with the antigen, multivalent antigen, or various antigens. A multivalent antigen has many types of antigenic determinants. When injected into the blood, each antigen determinant will stimulate the production of a particular antibody. The various antibodies produced by a single antigen combined with the different antigenic determinant. Okay, so we can also understand it by the formula, by the diagrammatic representation here. This is an antibody, and it is a antigen. If we talk about the affinity, if we talk about the affinity, this single antibody will bind to antigen, one antigen and two antigen, and their binding is very strong. Okay, so it is known as the affinity. One single antibody will bind to antigen, and the strength of the binding will known as the affinity. Similarly, if one single antigen and two antigen, one single this here is a antibody which is bound by two antigen epitopes. These are two epitopes, but their binding is not good. So one or two antibody will come and make the overall binding very strong. Here two more antibody are here which will bind to two different antigenic determinants, which make the it a very strong affinity, which make the overall affinity very strong. That is known as the avidity. Okay, so we can understand this by using this diagram. So now the next term, which is known as the cross reactivity. What is the cross reactivity? Cross reactivity. So we know that one antigen will be bound by a single type of antibody, but if sometimes one antibody will bound to non-specific antigen, it may be happens. It may also happen that one antigen which is specific to a particular kind of antigen may sometimes react to the similar antigen of another type is known as the cross-reaction. This is called the cross-reaction, and the antigen which produces the cross-reaction is called as the cross-reactive antigen. But the strength of the antibody released against its own antigen is strong. The bonds involved in the cross-section are weak. For example, if we talk about an example, the serum which is raised against albumin of an egg can a blood serum which is raised against albumin of hen egg can cross-react with the albumin obtained from the duck egg. So hen egg and duck egg, two antigens, two different antigens, but little bit similar antigens. So the serum which is raised in the duck hen egg can sometimes cross-react with duck egg. How it reacted to the duck egg? Because the duck egg little bit similar to hen egg. So this similarity uh, could enhance the binding of the serum to the duck egg, and the anti-serum raised in the human insulin can sometimes react with the insulin of pig, insulin of sheep, insulin of hell. Okay? So insulin is similar, but different hosts are there, but sometimes they are very similar, and this can lead to the binding of the antiserum or antibody to the that particular kind of antigen. And sometimes pneumococcal polysaccharides will react to the E. coli blood collection antigens also. So these are all are the examples, and we can understand it also with the help of diagram. This is the diagram by which we can understand. This is antigen, this is antibody, and this is antigen number 1, and this is antibody, and this is antigen number 2. The structure of antigen 1 and antigen 2 is similar, and the, this is the third number antigen. So this antigen number 1 which is similar, and this antigen number 2 which is similar. This antibody sometimes can interact with another antigen. So this is the original antigen, but this antibody should contact to this antigen only. But due to the similarity, this antibody could react with the antigen number 2 and antigen number 3. So this is the cross reactivity, and due to this cross reactivity, an antibody can bind to another antigens also. So this is all about the this is all about the antigens, and students can know about the cross reactivity, affinity, and avidity, and they can understand what is the affinity, what is avidity, and what is the cross reactivity. So the questions can be asked that uh, explain affinity, explain avidity, explain cross reactivity. Similarly, the questions may be combined with each other, which can uh, can I ask the question that explain affinity, avidity, and cross reactivity with suitable examples, with suitable diagram. So this is all about the affinity, avidity, and cross reactivity and antigenic properties. The lecture was about the antigenic properties and type of antigen antibody reactions, properties of antigen antibody reactions. So mm, uh, this is all about that. Thank you very much.